Alright, so for my biology role model project, I did Jennifer Doudna. A little bit about her background. She was born in Washington, D.C., which I'm pretty envious of. I've been to D.C. before, and it's really cool. She moved to Hawaii at a pretty young age, and she learned to love science in the rainforest near her home. Since she was little, she didn't really know what kind of science, but when her father gave her a biochemistry textbook, that really showed her what science she loved and what she wanted to pursue. I couldn't find much about her hobbies, and all I could really find about her personal life is that she has a husband named Jamie, who's a professor at USC Berkeley, and a son named Andrew. Although I haven't taken a biochemistry class yet, I really like bio, and I'm ashamed to admit that I really like chemistry as well. So I think that I have a strong interest in biochem, even though I'm not majoring in it, but I really look forward to taking my first biochem class, and I'm sure that I'm going to love it. Education. So she got her undergraduate education at Pomona College in California. It was small and had a strong biochemistry program, which is why she loved it. She also wanted to be close to home, so she tried to find something on the West Coast, so Pomona was a perfect fit. Graduate school was done at Harvard, which is pretty awesome. She studied RNA in Jack Szostak's lab, and together they helped engineer a self-replicating catalytic ribozyme. And this discovery furthered the hypothesis that RNA is the early basis of life, or the basis of early life, which she also does a lot of research on RNA to this day, and that is actually her main focus, but we'll get into that a little later. Her postdoctoral was done at the University of Colorado and Yale. So here we are to research. Her main focus is on something called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, kind of a mouthful. It is the hallmark of a bacterial defense system in genome editing, and in order to do the work on CRISPR, she moved to UC Berkeley, where her husband is a professor, like I said. To continue this work, she met her partner in research, Emmanuel Charpentier, at a conference in Puerto Rico, and they decided to work together to further this research on viral defensive bacteria. Jennifer actually received a Nobel Prize in Chemistry in 2020 for the research she did on CRISPR. Her most recent paper is called Precise Transcript Targeting by CRISPR-CSM Complexes, and it focuses on how CRISPR-CSM has a major advantage over CRISPR-Cas13, which was the most recently discovered CRISPR before CRISPR-CSM was discovered. This advantage is due to the fact that CRISPR-CSM uses a programmable RNA-guided mechanism to find and degrade target RNA molecules without inducing indiscriminate transcleavage of cellular RNAs. And the experiments done were done in zebrafish embryos as well as human cell cultures. And for my data figure, I have this little guy, which shows relative GFP fluorescence in cells transfected with Cas13 versus CSM, as well as the indicated GFP targeting cRRNA, measured by flow cytometry. And these lines here are error lines with standard deviation. And for my references, I made sure to put my paper in ACS format, and I have links for both the other websites as well as my images. Thank you.